So we're gonna finish up chapter 13, I hope. Now we're gonna focus on the DNA structure and how it's replicated. So I think we've seen these structures before, but now we get to spend a little more time with them. So DNA is composed of nucleotides, and there are 4 different nucleotides. They differ in their nitrogenous bases that are out here. The common part that they share is a phosphate and a deoxyribose sugar. It's deoxyribose if we're talking about DNA, and the deoxy, or the missing oxygen, is right here. If it's ribose, it has an OH in both places. If it's a deoxyribose, it's not no oxygen there, it just has a hydrogen. Very few differences between DNA and RNA. That's the one. Also, if you're talking about RNA, the thymine is a uracil, but let's focus on DNA for right now. So DNA consists of a nucleotide that consists of a phosphate, sugar, and a nitrogenous base. There are 4 different nitrogenous bases, and I hope you can notice the different shapes there. We'll talk about them more. So the structure, I told you about in the previous screencast that Watson and Crick proved that it was showed that it was a double helix by building models, but they confirmed their models, or they had the idea to build it as this double helix because of the data that was collected by Rosalind Franklin. She did this in the late 40s, early 1950s. She was a skilled crystallographer, meaning that she would purify things, make crystals out of them, bombard them with X-rays, and then look at the patterns to figure out what their structure is. A major feat in the early 1950s. Not only did she do a ton of work and create this famous X, which shows that the molecule she's doing this on, the DNA, has a helix pattern. She ended up dying because of her exposures to the X-rays. At the time, they didn't realize that you needed to be protected more from all these X-rays, and she ended up having ovarian cancer because of her exposure and died before they gave the Nobel Prize for the DNA structure, which is why Watson and Crick got the Nobel Prize for DNA structure. And Rosalind Franklin also had a collaborator named Wilkins, another man. He's actually the guy who shared the stuff with Watson and Crick. Franklin and Wilkins were not buddies. They reluctantly worked side by side. And he ended up getting the Nobel Prize after she died too. So anyway, she did this X-ray crystallography structure. The pattern here suggested that there was a repeating pattern that looks like a helix. And this is the familiar model that we always look at. A couple of cool features about the model. You can see it kind of in the center here where we don't have it twisted. Again, we've got our phosphate sugar base. And then interestingly, I want you to notice the directions. So this side, it says 5', so it goes from phosphate, sugar, and so on. And it ends with this last nucleotide, phosphate sugar base, that has a free OH on the 3' end. And basically, this guy is the one who's going to accept the next nucleotide if I was building a chain. We will continue talking about this as we talk about the enzymes that copy DNA. We did a lab way in the beginning of the school year about melting temperatures of DNA. And if you remember correctly, it takes higher temperature to break those 3 hydrogen bonds between the G's and the C's than the temperatures it requires to break the A's and the T's. So we were actually directly measuring this feature of the DNA molecule that's held together by hydrogen bonds between the nitrogenous bases. A's and T's make 2 hydrogen bonds, G's and C's make 3. And to their credit, Watson and Crick were the ones who did all the kind of model building to figure this out and notice that these molecules were available to do that bonding. Now, if we go and we twist the molecule together in that repeating helix, the double helix, it's not it actually has some texture to it. So it has what's called the minor groove. You can see this is closer together than the major groove up here. And that's a feature of the double helix, again, that they were able to notice from Rosalind Franklin's X-ray structure. And if we do a space filling, I think you can definitely see it here. We've got a minor groove and a major groove and a minor groove and a major groove. I think we talked about all this. So another thing to look at in this, and this was the point that Watson and Crick made, is that the spacing between the two strands can be perfect if you always pair a purine with a pyrimidine. And that's in this picture here. If you put two pyrimidines together, those are single rings, it doesn't get wide enough. If you put two purines, it's too wide. And if you do it this way, it's Goldilocks just right. And it's consistent with the X-ray data. Did that. So Watson and Crick in their 1953 structure paper are always noted as saying a very coy thing at the end, is that without any data at all, experimentally, they said that this double helix model can predict the way DNA is copied. And yeah, they were right. There were a series of experiments done called by Meselson and Stahl. They also won a Nobel Prize. They proved that the parent molecule of DNA separates, and then each of these parent strands will become the template for the new strand. This is another experiment that I'm not going to ask you to know the gory details of. I just need you to know that the term for this kind of copying is semi-conservative. Semi-conservative because you save half of the original molecule and you put the other new half on it. So now let's talk about the enzymes that are used to separate and copy the DNA. So I feel like I'm missing. Let's go to this picture first. So this one side by side is how an E. coli chromosome, which is a circular chromosome, opens up and copies itself into a new one. So there's a couple vocab words here. So there's always this thing called the origin of replication. That's where this is happening. We're going to take the parent strand and then create the new daughter strands. And this area where it's happening is called the replication fork, because that's where you kind of see a fork where the two sides of the DNA are being split and copied. If you're talking about a eukaryotic cell, we do not have circular DNA, we have linear DNA. So we can have multiple origins of replication, and then we have this replication fork going in two directions, and then eventually make two daughter molecules. So we're going to talk about all the enzymes that are involved in doing that. So the very first one that's going to have to happen is there's this enzyme called topoisomerase. And what this guy is there for is to help unwind a balled up piece of DNA that gets twisted because all this is happening. So he's hanging on to one side of the DNA. Then there is the helicase enzyme, which is the one that pops on and separates the hydrogen bonds and opens up that replication fork. Then the other proteins that are really important, we don't call these enzymes, they're just proteins, they're called single-strand binding proteins. So these guys latch on right after helicase to keep this fork from closing back on itself. Those hydrogen bonds really want to form, so we need to keep that separated. And then the last sort of getting everything started enzyme we need is called RNA primase, or it's called primase, and it makes an RNA primer. And this is an important little feature about the enzymes that evolved to copy DNA. So the enzyme that we're going to talk about in a minute is called DNA polymerase, and DNA polymerase requires a little something to hang on to. So remember I showed you that 3' end in the previous picture, the OH that's hanging off the nucleotide. Go backwards and look at that if you need to. So that 3' end is going to be given to RNA polymerase using this RNA primer. This guy's hanging out here ready to get attached to. Oh, I have it here. So remember I said there was the 5' end where the phosphate is. Then we have the sugar. And now we have repeating. Let's get down to the bottom. So here we got our phosphate, our sugar. And then the OH is the 3' OH hanging on to the end. So what's going to happen is that a new nucleotide is going to come in. Since this is an A, it's going to attract a thymine, And thymine is going to come in as a triphosphate. So remember ATP, and there's energy available when we break these bonds. So basically these nucleotides come in with their own energy to fuel this reaction. So these don't happen without any help. We have DNA polymerase, the enzyme that's going to help make that bond. And basically DNA polymerase is going to cut that bond right there and help this phosphate attach to the OH. The H is going to leave, and we are going to lose two phosphates and make that bond. You only can grow at the 3' end. So because you can only grow at the 3' end, there's one strand of the DNA that gets a primer, gets DNA polymerase attached to it, and is chugging along, making a nice piece of DNA. He's very happy and is making a nice long strand of DNA. Helicase is continuing to unwind at the fork. The single-strand binding proteins are holding it apart, and DNA polymerase is just chugging along, continually elongating from the 5' to the 3' direction. But what about this other side? And you can see it in this picture. The other side, and so that one that's chugging along just nice is called the leading strand. The one that is getting these breaks in it is called the lagging strand, because on the other side of the DNA, remember, you have to have a primer going 3' to 5'. But this guy can only keep adding when this opens up. Imagine this opens up some more, you can make a new primer, make another guy there. And they make these little bits. So these are called the lagging strand because it takes just a little bit longer to do that. In class, we'll look at a video together and talk about it. So why is that happening? I usually have to draw this on the board, so I don't know how much I'm actually going to end up talking about it here, but let's just take a peek at this picture. So we're going to zoom in on this part right here. So I told you that when the fork opens up, we have to keep making primers and keep elongating. So this one would have happened first, then this one happened, and now this one is going on. So if the primer is making a little primer, it can then go that far. And then it's going to have to go and make another one and go that far. And there's this little gap because these guys cannot connect to that one. Because remember, polymerase only goes 3' to 5'. This is 5', so it cannot attach to this guy. So what we have is this lagging strand, is these bunch of little pieces that were noticed by a scientist named Okazaki. So he called them Okazaki fragments. And he realized that there's another enzyme that can come in and fill that gap. This guy knows how to make that connection. And then there's an enzyme called DNA ligase that can seal up that little nick. Again, we're going to go through this in class, but I just want to make a quick pass of it here. So again, there's a lagging strand because polymerase only goes 5' to 3'. Then it butts up to the next one that was already there. Because there's this little gap, it needs to get filled in and then sealed up with ligase. Another kind of picture of it all happening, going through. So this would have been the first one. This is the second piece, this is the third piece, the fourth piece, the fifth piece. Every time that zipper opens up on the lagging strand, we have to make a new primer. DNA polymerase has to grab onto it, make a new copy, bumps into the old one, boom, falls off. So another little thing we didn't talk about much is that this little RNA primer ends up falling off. RNA is not supposed to be attached to DNA, and we have enzymes that remove that. And that's what they're showing you here. Another picture of the same thing. So I put in here a list of the big replication enzymes. And I would like you to know this, except that I don't need you to know the difference between these Pol 1s and Pol 3s. Helicase again unwinds the DNA and breaks the double bonds. Single-strand binding protein, technically not an enzyme. It binds and stabilizes the single-stranded DNA. Topoisomerase is going to be there to help that twisting. Imagine a telephone cord. You keep twisting as you're turning the other side of it. We'll demonstrate that in class. Primase is the enzyme that makes a little 5' RNA primer that's required to have DNA polymerase bind to. The Pol 1 is the one that removes the primer and fills in the little gaps. And then DNA ligase is the one that's going to fill in the gaps where the Okazaki fragments were. So here's that whole summary. We're going to watch a video that shows how this happens, but I'm not going to talk about it right here. Couple of little details. So DNA carries with itself a proofreading mechanism. Different DNA polymerases have different qualities. I'm going to speak with them in a generality, but there are different DNA. You saw Pol3, Pol1, different ones that have different properties for proofreading. But if there's an error, so the wrong nucleotide was added up here, a thing called a nuclease detects that there's a bubble and it will clip it out. It doesn't actually know which side is the wrong side. So this is how a mutation could get cemented into the genome. So there's a 50-50 chance it's going to pick out the wrong side. Then there's a special DNA polymerase that goes in and fills up that gap. And then we have DNA ligase to patch it. So as DNA is being copied, this whole slew of other enzymes are coming in there for proofreading. And that's why we are not riddled with mutations or changes. We have just enough to allow evolution to take place. And then lastly, I want to tell you about telomerase and the telomeres. So because of that 5' to 3' thing that you can only add to the 3' end, it ends up that you can't copy the very ends of the chromosomes. And that's what these little orange dots are. And I don't have a figure from the book that shows that. So I ended up putting in this video, which you can go and watch later. But remember, they had the lagging strand. And in this picture here, you can see that that RNA primer was removed because I told you RNA primers can't stick around. Well, the other thing our cells don't like is naked single-stranded DNA. So if there was no special thing on the end, your cells would be cutting off this little piece of single-stranded DNA, and your chromosomes would get shorter and shorter and shorter. And in fact, we see that during our lifetime. If there's not a patch there, your chromosomes get shorter and shorter and shorter. And they actually proved in yeast that this could cause the yeast to die. So when the tips are gone from the chromosomes unwind and the yeast cell will die. They think that also is going on in human aging. So how do we deal with the fact that these tips are going to go away? And there are some cells that need to replicate a lot. Think embryo. And then on the flip side, think about a cancer cell that wants to stay alive. So there is an enzyme called telomerase. And this telomerase acts on the edges of the chromosome. And these edges are repeating G's and T's called telomeres. So they often talk about these as those little plastic edges on your shoelaces, epilepts or epilepts, something like that. Anyway, think about these as that. These are junk DNA sequence, throwaway DNA sequence that's tagged on to the end of a chromosome. And then if these cells are supposed to be there, there is a special enzyme that recognizes it and fills in that gap. So if you're a cell that needs to copy itself a lot, let's talk about an early embryo. It has an active enzyme called telomerase that basically maintains the edges of the chromosomes. As a cell ages, telomerase is not expressed, and those telomeres are able to shrink down. Hopefully, they don't shrink before that cell dies. So you can watch this video on your own. You can go link to it from our main agenda webpage and watch this later. And I think, so I talk about histones. This was too long already. I'm going to stop. We'll talk later.